Hello, this is Professor White, and I will be discussing about genes and central dogma. Before we start, I just wanted to bring this to your attention about this mutation. This protein right here is called the CFTR protein, which is the one in green. This is embedded in the cell membrane. Now, changes in this protein can lead to what we call a cystic fibrosis. Now, we will explore today how this protein can become altered. And again, it lies in the DNA. So let's take a look first. One gene is one protein. So this is kind of like the hypothesis they have, that genes are the recipes for proteins. And proteins are the building block of enzymes. And enzymes are protein machines that actually carry out drug metabolism, for example, as a function of these enzymes. So we'll take a look at this today. So there are three important points to remember. Chromosomes are made up of DNA. The segment of DNA codes for a protein. And protein in turn relates to a trait, like color, enzymes, hormones, hemoglobin in red blood cells, and so on. So we are going to keep on referring these as we discuss our main lesson today, which is the central dogma. So the central dogma is very easy, kind of like an overview. DNA, RNA to protein. The double helix, which is your DNA, contains instruction. And that specific instruction is transcribed or copied over by the mRNA and translated at the ribosome. And in the ribosome, it is translated to proteins, depending on what instruction the DNA wants the cell to do. So going back to the structure of the DNA, we discussed this about the DNA. The DNA is a double helix, and it is double-stranded, and it has adenine, thymine, guanine, and cytosine as your nitrogen bases. So, and it's anti-parallel and so on. RNA is like DNA, except RNA has ribose instead of deoxyribose. It doesn't have thymine, but instead it has uracil, and it's single-stranded. And there are three forms of RNA: mRNA or messenger RNA, tRNA or transfer RNA, and rRNA, which is ribosomal RNA. So this is our RNA. So during the transcription process, this is where mRNA will be made from the DNA. Now, this follows the base pair rule, wherein uracil instead of thymine is replaced, and uracil will now be partnered up with adenine. So here's basically how the RNA is made from the DNA. Okay, so in the translation process, however, this is where protein is built from the mRNA that came from the nucleus. And the tRNA is used to build an amino acid chain, and each three bases of mRNA codes for a single amino acid. So translation occurs in the ribosome. mRNA is threaded through the small and large subunit in the ribosome. Now, each transfer RNA delivers an amino acid which binds to the previous amino acid, eventually forming a long chain of protein. So that's basically what is happening whenever your mRNA get together with the transfer RNA, and tRNA is actually the ones reading codes, 3 codons at a time. So this is a much closer look. So during translation, or we also call that protein synthesis, it always starts with an AUG sequence, which is the start codon. And as soon as the ribosome and your tRNA reads that, then it'll start the translation process. And it attaches to anticodon UAC. Once the start codon AUG start, of course, AUG pairs up with UAC, and it codes for a certain amino acid. In this case, that's methionine. Okay, so let me show you this video real quick on how DNA is translated to protein. As you can see here, that is your transcription process for making your mRNA, and it's going to get edited before it goes out of the cytoplasm to meet up with the ribosome. And the ribosome, together with the tRNA, will read the codes and provide 3 codons so you can produce your amino acids and protein. All right, so that's basically how protein synthesis happens. Here's a quick animation of what happens during the protein synthesis. This is a translation process right here. Already the tRNA is applying the codons. This is the transcription process. mRNA is made into DNA. RNA goes out, then translation happens in your ribosome. As we mentioned earlier, every 3 bases on mRNA is called codon. Now, each codon specifies 1 amino acid. Now, chains of amino acid become proteins. So, for example, in here you have lactase, catalase, CFTR, which was the protein in cystic fibrosis, insulin, hemoglobin, dystrophin. These are all proteins. And of course, it relies on the codes that the proteins made from, of course, the instruction coming from the nucleus in the DNA. And this is basically the 3 codes. If it's U, if you look at our first base, second base, and third base, then it codes for phenylalanine. If it's CCA, that's your proline. So that's basically how you read and how the amino acid is named in this particular chart. So going back to our phenomenon on the cystic fibrosis. So the cystic fibrosis protein is actually made due to wrong production of protein because of a frame shift mutation. So as you can see over here, because of that wrong protein, what happens is that protein that is made causes the lungs to produce extra mucus in the lungs of a person with cystic fibrosis, including their intestinal linings as well. And it has to do with a shift in the reading frame for a person with this genetic mutation. There's what we call regions of DNA, which is exons and introns. So recall in the transcription process, before the mRNA is sent out to the cytoplasm, the mRNA has some sections that are non-coding regions, so which is kind of like not really important. So spliceosomes basically removes those introns from the pre-RNA, and so that you will be left with the exons, which are of course the coding regions of the DNA that will be sent out to the cytoplasm for protein synthesis. So here's another diagram of what happens. So the DNA has those sections that are coding and non-coding region. Now during the transcription process, those introns are removed and the exons spliced together, and then that's the only coding sequence that needs to be sent out to the cytoplasm. The intron removal by pre-mRNA splicing occurs in the nucleus using the spliceosomes, like I mentioned earlier, and these molecular machines consist of small RNA molecules and associated proteins called SNRNP, or small nuclear ribonucleoprotein. Now let's go back to our phenomenon earlier, which is this protein, CFTR gene protein. Now, an error in what instruction coming out from the DNA happens, the gene sequence that comes out, and it's an error, can result, unfortunately, to mutated protein that can lead to cystic fibrosis. And also another mutation in the changes just with just a single DNA nucleotide is very evident in the mutations in the LMNA gene, which cause the Hutchinson-Gilford progeria syndrome. This gene provides instruction for making proteins called lamin A, which plays an important role in determining the shape of the nucleus within cells. Now, the mutation results in the production of abnormal versions of lamin protein, and the altered protein makes nuclear envelope unstable, making the cell more likely to die prematurely. In effect, causes senescence. It's like premature aging, as you have to say. And specifically, the mutation replaces the nucleotide cytosine with the nucleotide thymine in a certain position in the gene, and which caused this mutation. I hope this helps with your basic understanding of the genes and the central dogma. Again, this is Professor White, and I will see you in class. Bye.